Hello students, the topic we would learn today is ANOVA, that is analysis of variance. It is also known as F distribution, the name given as a tribute to the developer F.A. Fisher. We have already finished with variance and you are now familiar with what variance is when we talk about quantitative traits and how is it calculated. ANOVA is the analysis of these variances. ANOVA can be defined as a collection of statistical tools which helps us to analyze the difference in variance between the groups and within the groups. It helps us to conclude whether our experimental results are really significant or due to some error or by chance. The total variation is split into two components, variance between samples and variance within samples. One should remember if we have just two variables, we can go for t-test. This too you have practiced in your class. If there are more than two groups, then we go for ANOVA. That is, we are replacing multiple t-test with the ANOVA test. Now, when we do an experiment, there are two types of variables, an independent variable and a dependent variable. An independent variable is a variable that a researcher or the investigator can manipulate. The dependent variable depends upon the independent variable used, that is, in other words, the response to the independent variable. Now, based on the number of variables used, ANOVA can be one-way or two-way. We'll concentrate only on one-way ANOVA. Now, while doing one-way ANOVA, there are certain assumptions that are made. Firstly, the data of a dependent variable has normal distribution. That is, it is a continuous variable and has a bell curve. Second, the population is homogeneous or the variance and the standard deviation of the population should be equal. Lastly, the samples drawn from the population are at random and independent. Now, this slide summarizes the difference between one-way ANOVA and two-way ANOVA. If you say one-way ANOVA is a test to compare between means of three or more groups of a data, whereas two-way ANOVA is a comparison between the means of three or more groups where two independent variables are considered. You should remember for one-way ANOVA there is only one independent variable used, while for two-way ANOVA there are two independent variables involved. Now, let's understand it with an example. An experiment was conducted to check the weight loss in individuals based on diet and exercise. The volunteers were divided into three groups. Group 1 did regular exercise. Group 2 had a strict diet plan with an intention to reduce weight. And Group 3 did regular exercise along with a strict diet plan. At the end of the study, the weight loss was noted. Here, weight loss is dependent on diet and exercise or both. So, weight loss is a dependent variable dependent on the independent variable, that is diet or exercise. This can be manipulated by the researcher. Hope you understood it. Now here again if you notice the numbers within the group, okay, the volunteers who went to reduce weight only by means of exercise. Now if you see carefully, in group 1 all the volunteers had regular exercise protocol, still variation is seen in weight loss. This is said to be variance within the group. Same you can notice for other groups also. This variation is said to be variation by chance or by an error. Now, just notice the value or weight loss between the groups. That is group 1 who did exercise, group 2 who did diet, and the third group both diet and exercise. Now here again you will see variation in weight losses. This variation is due to the different treatments that they undertook, and some could be by chance. Now ANOVA helps us to know whether these variations are really significant or just by chance. So the variability that we see in an experimental data is due to variance between the treatment and variance within the treatment. Variance between the treatment is due to the treatment effects and chance factor, while variance within the treatment is due to chance. ANOVA helps us to decide whether this variation is really significant or not. For this, the first step that we should take is make a hypothesis. First, a null hypothesis, which says that there is no difference between the groups that we have considered. Statistically, the means of all the groups are equal. Then we give an alternative hypothesis that at least one of the mean values are not equal to the others. That means there is a difference between the groups. After stating the null hypothesis and the alternative hypothesis, we perform the ANOVA test. In the ANOVA test, the first thing we need to do is calculate the F statistical value. Then compare it with the F critical value based on a significant level and degree of freedom. The F critical value is obtained from the F distribution table based upon the degree of freedoms. If our calculated F value is greater than the F critical value, we reject the null hypothesis. Now, F statistics must be used in combination with p-value, which is nothing but probability of occurrence. A large F value means something is significant, while a small p-value means all our results are significant. You'll understand this while we are doing the problems. Now, this is a representation of an ANOVA table to calculate the F ratio. If you see, it's last in the column. Now, to calculate the F ratio from the data, we need to calculate the sum of squares between the treatments, sum of squares within the treatments, and then calculate the F value. This we'll do in detail while we are doing the problems. F ratio is the variance between the treatment to the variance within the treatment. A significance test is done to estimate whether the observed difference between two or more groups is due to by chance or not. For F ratio t-test chi-square, we use the p-value. The level of significance is denoted by alpha, which we usually take as 0.05 or 0.01. If p-value is less than 0.05, it is said to be statistically significant. Now, if our calculated F value is greater than the F critical value, we reject the null hypothesis. This figure summarizes what we have already learned. From the sample data, the first step that we need to do is create a hypothesis, a null hypothesis and an alternate hypothesis. Then we'll do statistical test to find the F ratio followed by p-value or significance. Now, based on whether the F value is greater than or less than the F critical value, we will conclude whether to accept the null hypothesis or reject the null hypothesis. Thank you.